In this video, we're going to discuss the last two components of your final lab report or scientific paper, which are the introduction and the abstract. So our goals are to prepare, analyze, and critique introductions and abstracts of scientific papers and lab reports. Let's start with the introduction. So the introduction should include references to previous primary research. This is really where the bulk of those references come in. This is where you show your audience that you've done your homework, you understand the topic. And introductions really vary in length. Sometimes they're 1 to 3 paragraphs, like for a lab report, and other times they can be multiple pages if it is a longer scientific paper. You want to write this after you've completed the experiment, but you present it as though that's not the case, right? It goes first in your paper. And it's written in a mix of past and present tense. Past tense is what you use for what you or others have done previously. Previous work has shown whatever that previous work is. This is where you use that literature to help back up why you're doing your experiments. And then present tense is where you put established scientific facts. So for example, if I was writing about Alzheimer's disease, I would say in the present tense, Alzheimer's is an incurable disorder. That is a known scientific fact. You can look it up on Wikipedia. Now, for the next sentence, I would use past tense. Previously, our lab found that drug X improves patients' memories. Okay. Past tense for something that I or my group or other scientists have done, but it's still not established as a scientific fact. It's more just a recent experiment. The introduction we call V-shaped. So we're going to start broad and get specific. Unlike the discussion, which is triangle shaped, starting specific and getting broad. The introduction really is the focusing of the story of your study. Remember, this is a narrative. You're not just conducting a series of experiments in isolation, but you're trying to tell a story. So you want to convince the reader why it's important, and you want to convince the reader that you are capable of doing this work. You've done your homework. So the rough outline, you start with something broad and interesting. I'm doing a paper about Alzheimer's. I start with the fact that Alzheimer's is an incurable disorder that affects millions of Americans and is currently the 6th leading cause of death in the United States. Then this will help engage the reader. It explains why the study is important, why they should read this paper, why they should care about the work that you've done. You want to provide then a targeted background on both your independent variable and your dependent variable and your model if appropriate. So if I was studying Alzheimer's in mice, I would explain a little bit about Alzheimer's mouse models and what we usually do with Alzheimer's mouse models. If I was looking at the effect of a drug on the memory of that mouse, I would explain basically how we measure memory in mice and what has been known about this drug or other similar drugs. You want to focus it on the study at hand. So I wouldn't tell you every single thing that has been done about Alzheimer's in the world, right? I'm focusing it on this drug, memory in mice. You want to explain what is known, and then you present that, we call it a gap in knowledge. So previous work has found this. No one has been able to find a drug to help people. That's why I'm doing this work. You want to close by providing a very specific overview of the project. So what did you do? You want to include the specific purpose, not something like confirm the importance of my work or confirm the importance of a drug, right? But a specific purpose to determine the dose and the effectiveness of a drug on memory in mice. And then you want to end with your complete hypothesis with a prediction. You want to assume this drug is going to work at a reasonable dose, will improve memory, etc. You also want to determine the appropriate amount of background information, and this can be a challenge. Again, if you were to look up on Google Scholar or PubMed Alzheimer's disease, you're going to find like a million articles, and your goal in an introduction is not to summarize everything that is known about that topic. We call it a targeted background approach. You want to provide your reader with the exact details they need to understand your work and understand why it's important, but not extraneous background and extraneous details. It's not a literature review. You want to consult your discussion. So what kinds of things did you talk about in the discussion? You want to bring them up for the first time, introduce them to your reader in the introduction. You want to consider writing the introduction backwards. So start with that hypothesis and prediction and work up in that V. All topics included in your report should be anticipated in the introduction. So if you found out something when you did your experiment and maybe one of them showed no difference and you think it has to do with X, so say the drug I tested didn't really work, but I think it's because maybe the memory test was designed for rats, but I used it for mice. I want to bring up rats versus mice in my introduction. Don't include information that isn't recalled or discussed later. So you only want to bring up the pertinent details. Some extra guidelines. You want to make sure you define any terms or concepts a biology major wouldn't understand. Please define any acronyms the first time you mention them. Provide support for claims of facts. Write an introduction for the study you conducted, not the one you hoped for, okay? Say you hoped that this drug would change the world and it turns out it's not really going to do that. Write about what you have. Allowable to include figures that aid in explanation. This is like so you can include like a background figure, and most of the time those are considered a cartoon, right? Or if you're studying a particular animal, you can include a picture of that animal, perhaps. But you don't want to have data in a figure. Conceptual diagram or your hypothesis. Again, that's the introduction. Now, the last thing you write is the very first thing a person reads, which is the abstract. And the abstract is a glorified summary. It includes information from each of the sections, and it never has any citations, and it should be about 300 words or a little less. This is the first chance to convince your reader that your paper is worth reading. It's worth their time. And so what you're going to do is write 1 to 2 sentences about each of the sections of your paper. 1 to 2 sentences of background, your hypothesis and purpose, the methods, the results, the conclusion or discussion. Determining the level of detail here is difficult because you don't have enough space to really go into far depth, but you also don't want to just skim the surface. You want to catch their attention. So usually it's 1 sentence that's a little bit more broad and the next sentence is a little bit more specific. Do not copy and paste sentences from your paper. You can tell, and it just doesn't flow right. You want to be as specific as possible without sacrificing breath. And for the results sentence, you want to include some numbers. This drug was twice as likely to improve memory as vehicle control. But you want to be as concise as possible and limit the use of any background articles. This is really about your paper and what you found.